The macula is a yellow oval-shaped area near the center of the retina of the human eye. The bulk of the macula is subdivided into the umbo, foveola, foveal avascular zone, or FAZ, fovea parafovea, and perifovea areas. The fovea, located near the center of the macula, is a small pit that contains the largest concentration of cone cells, which enable us to distinguish different colors. This is responsible for our central high-resolution color vision. Sharp central or foveal vision only covers about 3 degrees of our visual field as our eyes are focused straight ahead, but it allows us to drive, read, and see detail sharply. It also allows us to make important judgments such as estimating distance and understanding detail.
The yellow coloration of macula, observable in the living eye only under filtered light, comes from its content of yellow xanthophyll carotenoids, which are derived from the diet. This coloration allows it to absorb excess ultraviolet light entering the eye and acts as a natural sunblock for this area of the retina. There is some evidence that these carotenoids protect the pigmented region from some types of macular degeneration. Early onset macular degeneration may refer to several types of macular degeneration affecting children from birth to age 7. However, the most common cause is a genetic disorder known as Best disease or Best vitelliform macular dystrophy. Best disease is an autosomal dominant genetic form of macular degeneration that usually begins in childhood or adolescence and slowly progresses to affect central vision. The age of onset and severity of vision loss are highly variable. Best vitelliform macular dystrophy is associated with an abnormality in the VMD2 gene, which has been mapped to chromosome 11q13. However, some affected individuals do not have the VMD2 gene abnormality, which implies that other genes may also be associated with the condition. Affected individuals initially have normal vision, but eventually experience blurring and reduced clarity, or objects appear to have distorted shapes, a condition known as metamorphosia. Best disease affects central vision, but usually not peripheral vision. It varies in severity, even among members of the same family. Some people with the disorder do not notice a decline in vision, whereas others experience significant vision loss, especially after 40 years of age. The degree of visual loss may also differ in each eye. Best vitelliform macular dystrophy is a rare disease, and the prevalence is unknown. This condition has been diagnosed in individuals of European, African, and Hispanic ancestry. Currently, there is no known treatment for Best disease. A gene has already been mapped to a human chromosome, and hopefully will be soon identified as to its function in the retina. Fundoscopic examination in AMD is significant for drusen in the early stages of disease. These drusen usually are confluent with major pigment changes and accumulation of pigment in the posterior pole. RPE often appears atrophic with an easier visualization of the underlying choroidal plexus. In advanced stages of dry AMD, these focal islands of atrophy coalesce and form large zones of atrophy with severely affected vision. Other signs of choroidal neovascularization include RPE elevation, exudate, or subretinal fluid. The presence of these symptoms may indicate that neovascularization is occurring and that fluorescein angiography may be indicated to evaluate the retina. The periphery of patients with AMD also often has areas of drusen as well as RPE mottling and atrophy. Low vision aids, including special lenses or electronic systems that produce enlarged images of nearby objects, may help people who have vision loss from macular degeneration make the most of their remaining vision. One recent innovation, the implantable miniature telescope, may help those with end-stage AMD gain back some vision. The tiny telescope is inserted into one eye, which then provides central vision, while the other eye provides peripheral vision. The telescope projects images over healthy areas of the retina. The IMT can usually be implanted by an eye surgeon during an outpatient surgical visit. After surgery, patients must participate in a structured vision rehabilitation program to learn how to perform daily activities using the device. Currently, 3 potential neuroprotective strategies are being investigated for treatment of geographic atrophy: ciliary neurotrophic factor, or CNTF, bromonidine tartrate, and fluocinolone acetonide. Because GA progresses over a period of years, long-term delivery of these agents is thought to be vital. Each of these 3 drugs is being investigated in patients with GA, using a different extended delivery technology. CNTF is an encapsulated cell technology. Bromonidine is a biodegradable drug delivery system, and FA in the Illuvian drug delivery implant. The Neurotech encapsulated cell technology, ECT, has been called an implantable cellular factory. The intraocular implant, designated Renexus, contains modified human cells that secrete CNTF for sustained controlled delivery to the retina. The capsule implanted into the vitreoretinal space in an outpatient procedure is capable of secreting the protein continuously for more than 2 years, as demonstrated by explanted devices. Unlike other drug implants, this device does not primarily store the drug, but rather produces it in situ. The immune isolatory membrane surrounding the encapsulated cells allows ingress of oxygen and nutrients and egress of therapeutic factors, but blocks the entry of immune system components. This results in levels of CNTF in the vitreous that are consistent over time and effectively achieve photoreceptor preservation. A Phase 2 clinical study of CNTF delivered via ECT in patients with GA reported that treatment with CNTF resulted in a dose-dependent increase in retinal thickness. Visual acuity stabilization was seen in 96.3% of those in the high-dose group, compared with 83.3% in the low-dose group and 75% in the sham-treated group. Among those with baseline BCVA of 20/63 or better, 100% of those in the high-dose group lost less than 15 letters, compared with 55.5% in the combined low-dose and sham groups. Both the implant and implantation procedure were well tolerated. These findings suggest that CNTF delivered by ECT protects photoreceptors in GA, especially in eyes with 20/63 or better vision at baseline.